My name is Alexis, and this summer I had the opportunity to work in Dr. Kim's red blood cell lab, also referred to as the RBC lab. In this lab, we investigated different underlying principles of microparticles and proteins that could be suitable for the cytoskeleton of a red blood cell. Currently, there is a growing need for RBCs, primarily because of donor shortages and consistent struggles with transporting and storing blood. Globally, transfusions are a requirement for surgeries, car accidents, cancer treatments, and more. However, donated blood is often insufficient to meet this demand. That's why artificial red blood cells are a promising alternative that offer a long-lasting, stable, and are universally compatible solution. In my lab, I specifically worked on expressing and producing two types of proteins: spike-tag proteins, which are binding proteins, and spike-catcher proteins, which are long, flexible proteins. It was important to find a balance of the two. The more rigid proteins help the RBC maintain structure and function, and it was also important to note that the more flexible proteins allowed them to travel through blood vessels. We did this by inserting a target DNA sequence into E. coli, as it can live in humans. We did this while optimizing temperatures at 30 and 37 degrees Celsius, and we also managed expression rates for IPTG amounts. We noted that the more stiff proteins produce best at 37 degrees Celsius, whereas the more flexible proteins produce best at 30 degrees Celsius. We also then combined the proteins that were optimized using spike-tag and spike-catcher ratios. We did this to see whether the proteins would form long chains suitable for the cytoskeleton of a red blood cell. We found that the higher order oligomers performed best. In conclusion, it was important to note that it was a balance of rigidity and flexibility that helped to create the cytoskeleton of the red blood cell. In conclusion, this research will show that fine-tuning parameters such as incubation temperature and expression rates will produce the optimal proteins that can emulate red blood cells. Thank you.